Thank you. I'm happy to announce the last talk by my co-chair, John Ellerman, Efficiency and Safety Results of the RewinD LB phase in 28 clinical trial of Neflamapimod in dementia with Lewy body. You know the name.
Thank you, Carl. So this is actually going to be two of us speaking. I'm going to introduce and provide the background for the clinical study results, and the results itself will primarily be given by the first author here, Steve Gomperts at MGH. So these are our disclosures. So Neflamapimod is this oral small molecule drug that targets the specific molecular mechanisms underlying cholinergic degeneration, and in preclinical studies reverses the cholinergic degenerative process in the basal forebrain. That's published. Published data also from our phase 2A clinical study demonstrates that Neflamapimod has a potential or has effects on both the cognitive and motor aspects of DLB, and that effect is most prominent in patients who do not have AD copathology, so a pure DLB population with AD copathology assessed by plasma p-tau 181. This is the phase 2B design of the phase 2B RewinD LB study, which was a study designed to confirm those phase 2A studies, phase 2A results, and accordingly enrolled patients with DLB by consensus clinical criteria, again, who did not have AD copathology as assessed by plasma p-tau level being below 2.4 picogram per mil. The study enrolled 159 patients across sites in the 42 sites in the US, the Netherlands, and the UK. The initial study results from the placebo-controlled period were reported at the International Lewy Body Dementia Conference in January. Those results were negative, actually surprisingly negative, in the sense that there were no apparent differences between Neflamapimod at a dose of 40 milligrams TID and placebo. That's the dose and the effect that which we had seen effects in phase 2A. As we dug into the data more deeply, a hypothesis emerged based on looking at the plasma drug concentration levels, that there was a technical issue with the specific capsules used in the phase 2 and the placebo control portion that led to effectively underdosing of the patients. The plasma drug concentrations were actually closer to the 40 milligram twice a day dose level, which in the phase 2A study had largely proved ineffective. Now, what you're going to hear about in our presentation today is results from the extension phase where a newer batch of capsules was introduced. So you'll hear both about old capsules and new capsules. The old capsules are the capsules that were in the placebo control portion that were several years out from manufacture. We know now there were some physical chemistry differences that led to lower plasma concentration than would have been expected with 40 milligram TID. Those levels were in fact corrected with the new capsules. We have data from the extension phase, which then sets up today's results, which for all practical purposes, we reran the experiment in that extension phase. And now comparing a group of patients who received the old capsules, which effectively acted as a control arm, against new capsules, which are achieving target plasma drug concentrations and can be considered as an active drug arm. And so intrinsically, we're asking two different questions out of these results. One is the reason for the lack of results in the initial phase due to the differences in plasma drug concentrations? And then after that, if that is the case, and there is a clear difference between new capsule and old capsule, the profile of Neflamapimod as demonstrated by the data with the new capsule group. So there were 55 patients. The new capsules were introduced over time in the extension phase. There were 55 participants who only received the old capsules. Those are groups 1 and 2 during the 16 weeks that we'll be reporting on. And then 94 participants who had varying durations of new capsule administration, including 46 who only received the new capsules. In the data and the way we analyzed it, at the 2 visits where clinical outcomes were assessed at week 8 and week 8, the participant was considered to be on the capsule they were at that point. So there are more participants at the week 8 standpoint who received old capsules, the majority, while at week 16, the majority were receiving new capsules. Because of the time the way the new capsules were introduced, it was not randomized. So it's important to look at baseline characteristics. In fact, the 2 groups are well matched, except for on the bottom, the percentage of patients who were below a cutoff that we had used previously in the phase 2A study to define AD co-pathology. There's a higher proportion in the old capsule group who were below that cutoff level. So we'll report, the primary is all participants, but we'll give you data on the patients who are below only the patients who were below 2.2 picogram per mil. To the extent that we could, we stayed with the predefined analysis approach, both in the protocol and the statistical analysis plan, but obviously the comparison now is not Neflamapimod against placebo, but the primary comparison is between old capsule and new capsule. We did, where we could, also go back and try to make comparisons back to the placebo as well. And in most cases, we're reporting 95% confidence interval, but as with the original design, with the CDR sum of boxes, the primary outcome measure, we'll give you p-values as well. And then the CGIC, you can't calculate 95% confidence intervals. And with that, I'll turn it over to Steve.
Well, let's see. This slide shows the results of treatment on the primary outcome measure, a change in the CDR sum of boxes during the extension phase of the study. And as you can see on the left, for all participants, the subpopulation who received the new capsules, showed in green, had a smaller increase in the CDR sum of boxes over the course of the study, the 16-week period of the extension, than those who received the old capsules. The confidence intervals and p-values are shown down below. And then on the right, you can see the subset of participants who had a screening p-tau 181 value less than 2.2 picograms per milliliter, and these results, persistent if anything, are stronger. Another key analysis pursued was to evaluate how the CDR sum of boxes changed from baseline in the initial phase of the study over the 32-week period to the 16 weeks at the extension phase. And for that purpose, an MMRM analysis was performed, which showed there was a lower rate of progression of the CDR sum of boxes for participants who received the new capsules, shown in green, compared to those who received no treatment. And on the right, you can see the subset of patients, again, who had a p-tau 181 value less than 2.2, and these results are certainly persistent and more striking. Here is shown clinically relevant progression over 16 weeks, comparing the 16 weeks of the initial phase and the 16 weeks in the extension phase. And this is defined, progression is defined as at least a 1.5 point increase in the CDR sum of boxes over 16 weeks, or leaving the study. And if you look on the left for all participants, you can see that new capsules are associated with a lower progression compared to old capsules in the extension phase. And looking on the right, in this subset of people with particularly low p-tau values below 2.2, the contrast is stronger, with new capsules now contrasting significantly, not just with old capsules in the extension phase, but also with placebo treatment in the initial phase. One of the key secondary endpoints of the study is to look at the Alzheimer's disease cooperative study clinical global impression of change. And in the initial phase of the study, the results are shown on the bottom right. There was no significant difference between placebo or Neflamapimod in the context of the old batch of drug. So this was now pursued in the context of the extension phase, particularly at 8 weeks where the data were available. And what you can see on the left for all participants is that new capsules were associated with a lower CGIC compared to old capsules. And on the right, again, for the subset of patients with low p-tau 181 below 2.2, this result persisted and, if anything, grew stronger. A within participant comparison of the ADCS GIC was also performed in participants who received placebo in the initial phase. Transitioning from placebo in the initial phase to new capsules was associated with a significant improvement in the CGIC, but that wasn't observed with transition to old capsules. Shown here are results for a variety of other secondary and exploratory clinical endpoints. The data are organized so that signal to the right shows benefit with new capsules, signal to the left favors old capsules, and you can see across these measures there's a tendency to favor the new capsules. This proved significant for the Dementia Cognitive Fluctuation Scale, as well as the ISLT recognition, which probes working memory. But it's of interest that all but the neuropsychological test battery itself tended to the right. With respect to treatment emergent adverse events, these were low and comparable between old capsules and new capsules. There was a trend towards lower incidence of falls in the participants who received the new capsules. Otherwise, these were really comparable across the two groups. In examining the TEAEs in the subset of patients with p-tau 181 below 2.2, this incidence of falls now became significant, favoring with a lower incidence of falls in those who received the new capsules. And this is, otherwise there was no difference across the groups. This is shown graphically here with new capsules in the extension phase, showing a lower incidence of falls compared to old capsules in that same extension phase, but also lower than placebo in the initial phase. So to summarize then, positive effects with Neflamapimod treatment with new capsules were observed on multiple clinical endpoints. There was improvement on the primary outcome measure, the change in CDR sum of boxes, both with respect to old capsules during the first 16 weeks of the extension and against placebo, utilizing all data in the study through to week 32 in the 16 weeks of the extension phase. There was improvement on the CGIC in comparison to old capsules during the extension, as well as in a within participant comparison to placebo administration during the initial phase. And there was improvement with new versus old capsules in the DCFS, the fluctuations measure, as well as the ISLT recognition test, which, as I mentioned, probes working memory. And there were positive trends on the NPI-12, the timed up and go, and the UPDRS motor subscale as well, which I glossed over for time constraints. There were greater positive clinical effects in participants with levels of screening p-tau 181 less than 2.2 picograms per mil as well. The old and new capsules appear to have a similar overall safety tolerability profile. And in the subset of patients with this low p-tau below 2.2, we also observed, as described, a lower incidence of falls with new capsules compared to old capsules in the extension phase, as well as comparing against placebo in the initial phase of the study. These clear differences in clinical activity between new and old capsules is consistent with the hypothesis that the initial phase findings in RewinD DLB were the result of not having achieved pharmacologically active plasma drug concentrations in that phase of the study. Once target plasma drug concentrations were achieved, Neflamapimod demonstrated a meaningful effect on clinical progression as assessed by the CDR sum of boxes and the CGIC in patients with DLB who do not have AD copathology as reflected in the p-tau measure. These findings replicate the phase 2A study results and demonstrate proof of concept for Neflamapimod as a treatment for DLB. And it's worth mentioning that the pattern of the effects across a full set of clinical endpoints evaluated are consistent with the preclinical results in which Neflamapimod beneficially impacts the basal forebrain disease process. I and CervoMed would like to thank patients, caregivers, and study investigators, and the clinical site staff involved, the clinical project teams, members of the DSMB, as well as NIH, and thank the audience for staying through this session. Thank you.
Yeah, thank you for the nice presentations. So are there questions?
Just a quick question, really exciting results. Was wondering, was there some way to blind the participants to which type of capsule they were receiving?
Yeah, luckily we were all blinded. No one knew whether we were getting old capsules or new capsules. We all believed we were transitioning to an extension phase of the study with an open label extension with active drug. And it was only post hoc that we learned about this difficulty as CervoMed dug into the phase 2B results.
Great. Did they look the same?
Yes.
Great, thank you.
Okay. If there are no more questions, I thank you again. And I would like to thank